Hello, everyone. It's a pleasure to have this opportunity to speak to you today. I'm Steve Matlin, LifeLink CEO, as Jessica has already introduced me. I'm going to take about half an hour or 35 minutes to go through our presentation, and then we'll be able to take any questions that you have. And I appreciate this opportunity. Okay, so telomeres, what are they and why are they important? Probably most of you already know what this is, of course, but maybe we'll just do a very brief review of the fundamentals of telomere biology. So telomeres are the protective caps which are found at the ends of all chromosomes in not only human beings, but interestingly in virtually all living things on the planet. So that gives you some idea of the importance of telomeres in their role in evolution and biology. And the role of telomeres are to protect the genetic material that's encoding for protein expression, our genes, in the process of cellular replication. As we all know, cells are continually dividing in our bodies. And as these cells divide, these telomeres serve to protect the genetic material in our chromosomes to ensure that we have perfect replication of our DNA. But unfortunately, the process of cellular replication is slightly imperfect, and these telomeres are gradually being whittled away as cells divide. And this is essentially the main factor in driving the aging process. So what happens when these telomeres become short? Well, when the telomeres become short, they send the message to the cell that the cell needs to halt dividing, and the cells enter into what's called senescence, or eventually into what's called apoptosis. And as we have a greater level of senescence in our bodies, we are losing the ability to generate new tissues, we're losing immune function, and we are gradually losing our capacity of our stem cells to reproduce. And all of this is creating DNA damage, as we see here in the chart, leading to cellular damage, and ultimately coming up to the level of organ and organ systems, contributing to the onset of virtually all chronic and age-related diseases. We focused on some of the main ones here: cardiovascular disease, of course cancer, problems of fertility, metabolic diseases such as diabetes, particularly type 2, arthritis, central nervous system diseases. Stress is also very important and can be affects our telomeres very much. And of course, stem cells, which we need to renew our tissues, are often affected by our telomere length. So all of these things really, our telomeric-centric view of the world, show that telomeres are really at the core of all of the major diseases that concern us. And in fact, there are over 20,000 clinical, peer-reviewed, scientific and medical articles about the role of telomeres as a cause and contributing factor in all of these diseases. Sometimes we talk to people and they say, well, is the telomere the cause or the consequence? Well, the telomere is the cause. Short telomeres are driving the aging process, as we're going to see in a bit more detail now. So what determines our telomere length? Is it genes or is it lifestyle and environment? Well, it's a bit of both. So we estimate that about 1/3 of our longevity is determined by genetics, how long our parents lived, how long our grandparents lived. So there is a genetic factor. There are people who have inherited very good genes and are genetically inclined to live longer. But over time, as we move from being a baby to an adult, what really makes the difference, not only in our longevity, but in our health span, which I think is probably at least or more important than just lifespan itself, are the environmental factors. And environmental factors include where we live and how we live. So, of course, it's not the same to be living in a very contaminated city, breathing horribly polluted air, than to be living in the countryside. We perhaps don't have so much choice over where we live. But the second part of this, which is our lifestyle, is probably the most important factor of all. So, of course, lifestyle embraces a large range of activities, from whether we have a sedentary lifestyle to whether we do exercise. And when we talk about exercise, I always like to emphasize that we're not just talking about necessarily having to go to the gym every day and be on a treadmill and sweating and lifting weights. There are a lot of people that don't like that they find that boring, or they don't have the discipline to do that. What we really want to focus is not having a sedentary lifestyle. This can be working in your garden. It can be going for long walks with your dog. It can be any activity that gets you away from what I think is a new disease that we're going to have to find a word for it, which is basically looking at a screen all day. Looking at your mobile phone screen, looking at your computer screen, looking at your iPad screen, basically being in front of a screen and not really doing anything physically for your body. And of course, then diet, nutrition is equally important. We'll talk about that more, as well as the importance of trying not to smoke, to take alcohol in moderation, and of course, managing stress. I think stress is one of the greatly underestimated causes of health problems and indeed of aging. And it's very difficult to measure stress. But high levels of stress, if it's not managed well, do significantly contribute to the aging process and to telomere attrition. And this can be mental stress, and it can also be physical stress. Of course, inflammation from obesity, for example, is a stress on your body. But equally so are having long-term situations where you have poor relationships, or you're unhappy in your job, or you have a sick child. All these things, of course, generate stress on you, and this impacts your aging process. So these are the factors that are pulling together that are being reflected in telomere measurement. And this is what we're seeing when we see a report at any point in time. And just to emphasize, as we said before, this is something that's now well established. Telomere biology has moved out of the world of scientists to clinical application. And here are just a small selection, a few of the articles from major journals like the Journal of American Medical Association, JAMA, or publications by universities like Northwestern University, and even the British Medical Journal, probably one of the most prestigious medical journals in the world, which put on the cover of the magazine, telomere length is an independent risk factor of heart disease. So this is something that really, as clinical practitioners, has gotten to the maturity that this would be a tool used in clinical practice, because it's giving you insight to the pathologies, to the diseases that your patients are at risk for developing, and affording you the opportunity to take interventions well before you're going to see it in traditional metabolic tests, which often really don't show any evidence of disease, really don't give you any advance warning of the development of disease until it's already onset. So what are some of the major causes of aging? As we said, we think telomeres are the most important factor, but there are other factors. But I think it's interesting to focus on this publication in the very prestigious journal Cell, published in 2013, in which they identified a number of factors, and you can see them here, ranging from cellular senescence, stem cell exhaustion, poor cellular communication, instability in the genome, telomere attrition, of course, epigenetic factors, loss of proteostasis, and deregulated nutrient sensing and mitochondrial dysfunction. How do these factors work together? Well, let's move forward in the slide, and we see here that, in fact, 4 of the factors are downstream from telomere attrition. So the senescence of cells comes because telomeres are critically short, and the cells receive the message that continuing to divide is going to potentially expose this individual to mutations, which can give rise to cancer, and so the cells stop dividing to protect the risk of having chromosomal aberrations. And the other factors that we see, genomic instability, that's also caused by telomere attrition, the poor intercellular communication as well, and of course stem cell exhaustion. Stem cells, as we all know, are the undifferentiated tissues, undifferentiated cells that precede somatic tissues that replenish our somatic cells. And short telomeres in stem cells means that any kind of stem cell therapy that we would want to do isn't going to be effective, because the proliferative capacity of those stem cells is going to be very limited. So looking at telomeres in stem cells is a very powerful tool for the characterization of the quality of the stem cells. And indeed, we work in that area, and if anyone has any interest in that field, they should let us know. So what about reversing aging? How can we go back to things? What would be some of the things that we need to do each of these damaging factors? Well, we'd like to clear out the senescent cells. We'd like to use stem cell therapies. We'd like to have drugs that help reduce inflammation. Of course, much better is to remove the causes of the inflammation to begin with, such as not being obese, having a healthy diet. We'd like to eliminate damaged cells. We'd have drugs, epigenetic drugs that can reprogram our genes. Caloric restriction, I think, is now a pretty important factor that people need to realize, that being overweight is one of the unhealthiest things you can do. The burden that extra weight puts on your body over time in a sustained manner is a very unhealthy thing, and so some dietary restriction is very good if we can do that. And of course, telomerase reactivation, because telomerase reactivation would then address many of these other factors. So of course, there's many projects underway in the pharmaceutical industry to look for drugs, molecules, or gene therapies that will allow for the upregulation of telomerase in our healthy somatic cells transitorily. Ideally, what we'd like to see is to be able to turn back the clock in our cells through upregulation of telomerase in the healthy somatic cells. And that would essentially turn back the clock in our bodies. It would eventually be rejuvenation of the organs and of the organism. Of course, what we don't want to do is we don't want to upregulate telomerase in cancer. I know many people are concerned about that. But in fact, that isn't really a problem, because cancer is by itself able to upregulate the telomerase enzyme, because in the process of mutation, the genes that regulate telomerase are turned back on, which is why cancer becomes immortal. So we really want to focus on upregulation of telomerase activity in healthy cells. And if we can, at the same time, one of the most powerful ways to combat cancer would be the suppression of telomerase in cancer cells. And indeed, there are many projects underway, again, in the pharmaceutical industry, looking to do just that. And of course, there are now many companies that are selling products that are nutraceuticals and supplements, and which aim to support healthy telomeres, and in some cases to cause expression of telomerase in healthy tissues. And some of these companies have some pretty good basis for their claims. But I think it's very important that you as physicians evaluate the clinical need of this patient, and of course evaluate very carefully to differentiate between the companies that have really good scientific basis for what they're selling versus some others that perhaps don't have all the clinical evidence that you'd like to see. So we talked a little bit about telomerase. Just to emphasize again, telomerase is the enzyme which causes de novo additions to telomeres. This is normally only found in our germline cells. Otherwise, babies would be born old, and indeed there are some rare diseases where there is a dysfunction in telomerase expression and causes such diseases as aplastic anemia or dyskeratosis congenita. You do have greatly accelerated aging. So we normally find it in our germline cells, in our stem cells, and of course we find it in cancer cells, as we were discussed. So basically, telomerase is a Jekyll and Hyde enzyme in which we want it to be active in our healthy cells, and we would ideally not want it in cancer. And this is because cancer cells need to maintain their telomeres to continue to divide. So cancer cells tend to have quite short telomeres because they're furiously dividing. Cancer cells escape the Hayflick limit. They don't have this limited capacity to divide as do normal healthy somatic cells. And because of this, this allows cancer tumors to grow very quickly. And this is the explanation why a virtually unperceptible tumor in the prostate or the breast or the liver or the spleen or any type of cancer, a solid tumor, can go from something that's virtually undetectable to a very large tumor in a very short period of time, because the telomerase enzymes active in cancer are maintaining all of those cells completely operational and able to divide indefinitely, essentially giving them immortality. Here we see that think the title of the slide is slightly off. What we want to show with this slide really is that telomere length is a good biomarker. And the reason it's a good biomarker is because telomere length is very heterogeneous in the human population. You can see here at birth, some people can be born as telomeres as long as 12 or more kilobases, and others with 8. So there's a huge range, more than 50% range, between individuals at birth. And you can see that there's a line there that's showing that over time, in a very large study of over 800 individuals, up to the age of 100 years, there is a slow and pretty much inexorable decline in telomere length. But what you can't see here is what is the path of each individual person. And that's because the individual path of telomere attrition for each individual is unique. There may be individuals, there will certainly be individuals in this data that were born with telomeres, very long telomeres, 11, 12 kilobases perhaps or more. But because of a sedentary lifestyle, because of stress, because of bad diet, because of obesity, they had very accelerated telomere attrition. And by the age of 50 or 60, they could have already had heart attacks, and they could have passed away. But other individuals who maybe have born with much less of a genetic inheritance, but took care of themselves, had very slow telomere attrition, and the slope of their line is almost flat, and they can live to the ripe old age of 80, 90, or even 100. And that is what makes telomere length a good biomarker. Because if every dot on this graph was on the line, then that would mean there's nothing you can do about it. Everyone's the same. The rate of attrition is the same for all of us. And there's really no point in measuring something in which you can't do anything to change. But we, in fact, know that lifestyle changes do modulate telomere attrition very significantly. And hence, this is a very powerful tool for being able to monitor the efficacy of lifestyle programs and to see how all of these factors that we're discussing are affecting our rate of aging. Now, I want to talk a little bit more about cancer and why telomerase and telomere dysfunction lead to a mortality in cancer. So normally, in healthy cells, you have a decline in telomere length over time. And for most cells, they will enter into this range where they become senescent, as you can see here on the graph, and they will stop dividing, essentially to protect our somatic tissues and not allow for any kind of mutation to arise. And this is on the one hand bad for us because we'd like to keep those tissues healthy, but what the cells recognize is because the risk of a chromosomal aberration is greater, they need to stop dividing. And eventually they get into a further state of decline called apoptosis, where essentially they're now dead and they're now hopefully being cleared by the body. What happens, though, in some cases, is that perhaps because of some genetic alteration, together with other environmental factors, we can think about being exposed to cigarette smoke, we can think about being exposed to too much sun in our skin, we can think about too much alcohol. These factors, perhaps with some genetic predisposition, will cause some cells which will continue to divide, will have a mutation, and they will then become pre-malignant tumors and then move on to malignancy in some cases, essentially turning back on these telomerase genes, achieving immortality, and then developing into a full-stage cancer. And I talked before about some of the work that's being done in this field to combat cancer. And I think this is one of the examples in the public domain that we can discuss, because we have many projects that we're doing for companies that are not public. But this is one example of a compound developed by a company called Geron Corporation, which is a biotech company in the Bay Area. And they spent many years trying to find small molecules that would inhibit the expression of telomerase. And eventually they got it to a point where it was sufficiently mature that Johnson & Johnson has taken an out license on it and is now running clinical trials on it. And essentially the mechanism of action is to impede the upregulation of telomerase in these cancer cells. And essentially that's like taking away oxygen from a fire. So if there's no oxygen, the fire will eventually die out. Well, if you take away the enzyme telomerase from cancer, eventually the telomeres then become critically short in the cancer, the cells stop proliferating, and the cancer will die out. So this is a very promising drug. It's now, as I say, in clinical trials, registered trials with the FDA, and we hope, obviously, all of us, that this is a successful drug because it would be the first drug of its kind to combat cancer through the mechanism of telomerase inhibition. And on the flip side, we talked about the benefit of telomerase upregulation in healthy tissues. And here, we're not quite at the level of clinical trials. We're still in the stage of doing work with laboratory models, which are mice. But mice are pretty good models for human beings because it turns out that telomeres in all mammals, of which mice are, of course, are a mammal, have the same genetic structure, the same sequence of nucleic acids, TT, AGG. And the only difference between mice and people in this area of biology is the length of the telomeres. Mice have telomeres that are approximately 10 times as long as human beings, and the rate of attrition. Telomere attrition in mice is approximately 100 times as fast as human beings. And that, at least in part, explains why typical mice live 2 years and people live 80 years. So here what they've done is they've used some gene therapy, some what are called vectors, to transfect the mice with an extra, with SNPs of genes that upregulate this component of the gene called the TERT. And this, what it does is it causes the expression of telomerase for a period of time. Interesting, it's transitorial, which is good because what we wouldn't want to do is have it turned on all the time, because potentially we could revert cells back to possibly to a pre-somatic stage. So here what they've been able to achieve is they've been able to show that the upregulation of telomerase in these mice led to mice that had increased lifespan and increased health span. And this has been published. You can see the references here. If you're interested in the articles, you go PubMed and get them. And if you don't have them and don't have access to them, we're happy to send them to you. This is very, very promising work. Of course, more work needs to be done to do this. We're not at a stage where we can do gene therapy on human beings, and at least not with the regulatory permissions that would be needed for wide adoption. But this is pointing to a pathway that it is possible to do this in healthy cells. So just let me take a minute as well just to give, for those of you who don't know perhaps all about our company, a little bit of background. This is a photograph of actually of our old labs. We moved last December into newer and even better labs. And if anybody is going to be in Madrid, you all have a standing invitation to just send us an email, and we're very happy to arrange a tour of our labs. We're very proud of our labs. We are the only laboratory in Spain to be U.S. federally accredited, CLIA accredited. This is something which is not easy to achieve, as you can appreciate. There aren't many laboratories outside the United States that are U.S. federally accredited. And we also hold a number of other licenses. We're accredited under by the Spanish Ministry of Health, of course. We're accredited under ISO 15189. Many of you may be familiar with the term of ISO 9001. These are accreditations that go to quality and procedure and ensure that companies have protocols in place for ensuring the accuracy and quality of their services. The 9000 series ISO is generic to any industry, but the ISOs that are 5 digits, in this case 15189, are specific to an industry. So this is the highest ISO that exists for clinical laboratories, and we hold that accreditation. And we're actually the only lab in Europe that's both US government certified and certified under ISO 15189. And our company began, again, for those of you who do not know this, as a spin-off of this prestigious Spanish National Cancer Research Institute. So we are very focused on quality, on good science, and virtually everybody that works in our lab are PhDs in molecular, cellular biology, genetics, or medical doctors. And of course, we think that we are really offering the best technology in the world. This is a unique and proprietary high-cut screening technology with a very high level of accuracy, precision, reproducibility. And of course, we're the only company in the world that's measuring the individual telomeres one by one. And this is important because what drives cellular senescence is the shortest telomeres in the cell. I think I'll talk a little bit about this in a moment further in the presentation. And just again, to give you some more information about us, these are some of the major academic studies in which our company has participated. These are major EU-funded projects. For U.S. doctors, these would be like the equivalent of NIH grants, perhaps even more competitive. And the most important of which is now ongoing, is our study for the validation of our technology, telomere analysis technology, or TAT, as a diagnostic tool, a biomarker for cancer monitoring and prognosis. This study, which we awarded last year, is for over 3 million euros. It's going on now. It's going to last from, it started in January of this year, 2017. It'll run to the end of 2018, 24 months. It's the largest study ever conducted in the world in cancer and telomere diagnostics. And the objective of the study is to demonstrate that our technology uniquely can guide oncologists to know what will be the prognosis, the development of specific cancers, and in order to allow them to take treatment decisions. So we're not trying with this to identify if someone has cancer or not. There are many good tests available for the determination of that. But what we want to do is we want to tell doctors how people are going to respond by using what is essentially a non-invasive test compared to what we have today. Because the majority of cancers, as we know, are solid tumors. Breast cancer, lung cancer, prostate cancer, the big cancers are also tumors. And it's very difficult to monitor these tumors because the techniques to monitor them would essentially involve either repeated biopsies, which is not feasible because of the risk of the patients, the requirement for anesthesia, the cost. So we can't biopsy people every few months. And of course, the other tools like tomography and imaging, CT scans, are also very expensive tools that can't be applied with perhaps the frequency that oncologists like, not only because of cost, but because they expose the individuals to radiation, which can give rise to secondary tumors. So oncologists many times are forced to give treatments where they really don't have insight into how these treatments are affecting the tumor, or if they're responding at all, until many, many months have gone by. Of course, during that time, these treatments are very, very expensive. They produce tremendous side effects in the patients, nausea, suppression of the immune system. Psychologically, of course, people are depressed. People lose their hair, all the different things that we know. And of course, the worst outcome of all of this is you go through all of these procedures, and then you're in that often significant percentage of the patients who don't respond. You don't respond to the standard protocol treatment. So what we need desperately in cancer are better biomarkers that can give the oncologist insight into which treatments are likely to have more efficacy, and we think our technology can afford that. So some of the cancers that we're focusing on, these are some of the hospitals. There are over 32 major Spanish hospitals. All of the most important hospitals, public and private hospitals of Spain, are participating in the study. And we've been through the ethical review boards of each of these hospitals. And I think it's important to highlight that every single head of oncology of every hospital wanted to participate in the study, and every single hospital that we've approached approved to participate in the study. So I think the field of oncology, it's a very tough field to be a doctor in oncology, to be an oncologist. But the level of response that we saw from the oncology community is very important, because it suggests that the oncologists also share this view that this technology, this biomarker of ours, can give them insight that will allow them for better patient care. And the cancers that we're focusing on are primarily lung cancer in men and women, prostate cancer in men, as well as leukemia, both in children and in adults, and some other sophisticated areas such as the impact of immunotherapies on cancer. We're looking at immunotherapies across a number of types of solid tumors. There are going to be over 4,000 individuals between patients with cancer and controls participating in the study. They say this is the largest study of its kind ever conducted to date in the world, with a clinically validated and accredited high complexity assay such as the TAT. And these are just some of the data on the number of people participating in the study. We'll be analyzing something close to 7,000 samples. So it's a very, very large study, and it's going to show some very strong clinical evidence around how telomere biology can be used to monitor and determine outcomes of cancer and treatments. Now, I want to, as I reach the end of the presentation, just also highlight for you why it is that our technology is objectively the best technology that exists in the world today. And it isn't just because I say so. It's because of concrete science that underpins what we're doing. The technologies that you may be familiar with, or which are available commercially, are quantitative polymerase chain reaction, or qPCR, flow cytometry, and our technology, telomere analysis technology, which is based on an underlying technology which is called high throughput quantitative fluorescence in situ hybridization, or FISH. So let me just mention briefly what each of these technologies does and doesn't do. So PCR is a scalable technology. That is, you can run a lot of samples relatively inexpensively, which if you're a lab and you want to just get a lot of samples in and you want to price your test relatively inexpensively, that's good. The problem with PCR is there's a very high CV, or coefficient of variability. Unless it's done very rigorously, you can have you can take the same sample, run it twice, run it 3 times, and get 3 different results, with differences in some cases as great as plus or minus 20%. If we think about this in terms of an analogy that most of you will be familiar with, think about cholesterol testing. The standard threshold for high cholesterol is above 200. If the cholesterol assay that you were using with the same sample could be plus minus 20, that means you could run the same sample 3 times and get 240, 200, and 160. That means from the highest result to the lowest result, you have a 50% difference. From 240 to 160, you have a 50% difference on the same sample run 3 times. Clearly, you can't decide if you're going to give somebody a statin. If you have 240, you might think about statins. If you have 160, you're saying you're doing great. So that's not really an acceptable level of coefficient of variation in a clinical assay. And PCR, again, maybe possible to get those CVs down, but generally the standard protocols of PCR have a coefficient of variation on the order of 20%. In addition to that, it doesn't have much clinical value. We talked before that it's the shortest telomeres that cause cells to become senescent. What does PCR tell us about short telomeres? Nothing. It tells us nothing. All PCR does, it gives us an estimated average telomere length of the sample, not at the cellular level of the entire sample. So that really has no clinical validity in terms of looking at what level of senescence does an individual have, or what level of senescence do that individual's cells have. So my father used to say, that's like saying you've got one hand in the freezer and one hand in the frying pan, and on average you're comfortable. It's pretty far from the truth. So averages can be very deceiving, and that's why we think PCR has limited clinical value. Flow cytometry. Well, flow cytometry is a more robust technique, and it does give us a little bit more insight because it does measure average telomere length at the level of the cell. So now it's not the cell population, it's the level of the cell. But again, we still don't have that critical information as what is the shortest telomeres within that cell. We can't get that with flow cytometry. So this is just a limitation of the technology. It doesn't work, it's not detailed enough, it's not granular enough to look at the individual cells, the individual chromosomes, I'm sorry. And there's another problem with it, which is not very scalable, which means that as a tool for doctors, it's a very expensive assay to run, and it's very expensive to perform, and then it's going to cost you and your patients a lot of money, and therefore the commercial viability of this, for this to be widely adopted, is going to be limited. And then we come to TAT, or HTQ FISH, as the underlying technology. Now, in LifeLength's hands, we have been able to take this technology to a point that this is highly scalable. We can run hundreds of samples a day, thousands of samples per week. Thus, our assay has become very affordable. And in terms of the quality, it's by far the most accurate, reproducible, and precise technology that exists. And we have all the data to back that up. And on top of that, it gives clinical value because we're measuring from the individual chromosome, the individual telomeres in the cell, over 100,000 typically in a single sample, running all the samples in quintuplicate. And what this gives us is a level of granularity from which we can then determine what is the percentage of the short telomeres, what is the median telomere length, which is more insightful than average telomere length because telomere distribution is not a bell-shaped curve, but it's rather weighted to the shorter end with a long tail. If we think about the difference back to our days of math, there's a big difference between mean and median when the distribution is not bell-shaped. So we all know that medians are better representations of data sets when distribution is not Gaussian. And from that, of course, we can derive any piece of data you want. We can say, how many telomeres are there below this length? How many telomeres are between this length and that length? What is the median? What is the average? So we can do everything you can do with these other assays, but these other assays can't do what we do. And we do it with a level of reproducibility and accuracy that no one else has. Because this is all we do. LifeLink is a laboratory solely focused on telomere biology and telomere diagnostics. And this is just the evolution of the technologies that existed. It's been almost 30 years now since we had our first primitive assays with something called TRS, which were electrophoresis gels, to measure telomeres. Then we worked through other technologies to where we are today, which is really the state-of-the-art technology for telomere measurement. And this is just another visualization of what I was saying. So with PCR, you can see that you can basically look at average telomere length in the population of cells. You can run a fair number of samples, but again, it doesn't really give you much insight. When you move to flow cytometry, you're now seeing single cells, average single cell length distribution, again, on the average of the population, but still not a lot of insight into the shortest telomeres. And then when you move to our assay, TAT, this is where you get the finest level of detail, individual measurement by chromosome. And of course, if you have that, you have everything else. Now here's a little bit on our report. Everyone who's worked with us will hopefully have seen the report and read it and done your own test, of course, but if you haven't, you should do it. And I want to highlight how some of that data, how some of that technology transforms into clinically useful data. So these are a couple of the most important pages of the report. So on this first page, you have the median telomere length of this person's blood sample. And it's graphed on this horizontal bar, which goes from red to green, with about 95% of the patients falling between short, normal, and long, and only 5% between very short and very long. Now, what's important about this graph is it says below in the box, the range in this graph are dynamic and based on your age. And you say, what does that mean? Now, most of you will be familiar with standard lab reports that don't take into consideration a patient's age. Cholesterol testing, you don't have to know the patient's age for cholesterol. Blood pressure, you don't have to know the patient's age for blood pressure. So other risk tests, other screening tests, don't ever consider the patient's age. But in telomere biology, if you don't do that, you're going to have a situation where all older people are going to look bad and all younger people are generally going to look good. Because as we saw before, telomeres inexorably decline with age. The cells divide. The only question is at what rate do they divide? If you're very healthy, if you're very great genes, if you have a great lifestyle, it's going to be much slower than if you have all the other factors against you. But they're still going to decline. So if we simply had a graph that was static, you would inevitably find that the patients who are older are going to look worse than the patients who are younger. And that's not very encouraging. To compare myself to people who are half my age in a biological test in something that I can't control entirely doesn't make a lot of sense. So our database is big enough that we're able to give you a graph that is individual for that person's chronological age. And this is something that no one else does because no one else has the database that we've developed. And you'll see that we report our median telomere length and we report standard deviations. And this is another level of transparency in the quality of our test because every biological test has a standard deviation. Go find a lab report that shows you standard deviation around the accuracy of their assay. No one does this except us because there's a measurement error in any biological assay. And we're showing you that we have very low standard deviations in our test. And then we give you the average telomere length. And we said before that that's not very helpful. But the other companies that do telomere length can only report averages. So we provide it to you, because if you want to compare this with somebody else's essay, then at least you have the value. And then we say, where does that person fall in our database? And you can see here this individual is falling in the normal group, a little bit on the lower end. So basically 34% of people of this age have a shorter median telomere length, and 66% or 65% have longer telomeres. And you can see as a consequence of that, this person's biological age is a little bit older than their chronological age. Not dramatically so. And there is some coefficient of variation in our sites. We believe it's below 5%, not the 20% of PCR, but there's a little bit of movement if we rerun samples. But this is a person who's basically doing well. Not fantastic, but not bad. And then when we move to the right, we look at the 20th percentile. We talked before, again, how this is something uniquely offered by our company. The ability to look at the shortest telomeres, which are, if you will, the bad actors, the ones that drive cellular senescence. And while these two graphs on the left-hand side and the right-hand side are often the same, they're not always the same because the distribution is not Gaussian. So sometimes you can see people that have normal telomere length and median values, but have a much higher percentage of 20th percentile. But in this case, this individual has median telomere values and short telomeres that are basically healthy for his or her age. And you can see again where they fall in the database. So relatively speaking, he's better in terms of short-term telomeres because he's only 60% of people are better than him versus the 65% or so that we saw on the other graph. And these, again, both these graphs are dynamic for this individual's age. And then, of course, this is not a test that you would want to do one time. The clinical value of what we're doing is really being derived by doing the assay once a year for, ideally, many, many, many years. Because that's the way you can see the efficacy of the lifestyle program. I said before that we try to do things with a great deal of transparency, and I think it's important to highlight that we don't recommend people retesting prior to a year, but we strongly recommend they retest every year. And the reason for this is because this is not a metabolic assay like a cholesterol test or glucose or hormone testing, where you can see differences in a few days or weeks. And you can put somebody on a low-sodium diet and see those changes, or you can put yourself on a low-fat diet and see the change in cholesterol. We don't suggest that because telomeres do change, but they don't change day by day, at least not with a level that is detectable by anyone in the world, not even by us, not yet at least. But neither is this a genetic test. You know, you do a genetic test and you know your SNPs and you know you have your risk for a particular pathology, you're done. If the lab has done a good job, that test is accurate, you're not going to repeat that test. So telomere testing sits in the middle. And this is a test where the clinical value comes not only from the first test where you see where you stand against a population, a database, but over time. And here you see that this person over a year and a half or so is essentially maintaining their telomere length. There's a little bit of movement. That's because we're looking at white blood cells, so there can be some variation in the white blood cells. Maybe the person's had a cold recently, maybe they had stress. There is some movement there, but over 3 tests, you can see there's no real meaningful decline in the telomere length in this individual. So that's someone who's doing well. They're keeping their biological age essentially static. And if we were to go back and look at actually the first report, the gap between their chronological age and biological age was greater. So in the first test, they would have been biologically older, relative in terms of years, older than they are now. So now they're catching up to the point where if they continue, at some point, they're going to have a biological age below their chronological age, which is what we want. And then we represent this in the lower graph on this dotted line, on this line rather, where the line is basically the swing point. If you're below the line, you're older biologically and chronologically. If you're above the line, you're younger. You'd like to be above the line. And so this is really, I think they're good graphs and they give a lot of insight to people. And our test is set up for this. So when people retest, we simply ask them to give us the prior sample code, and this is all automatic. So it's really easy for you to track your patients over time. And the last thing I want to highlight is our online questionnaire. If you've worked with us, you know that we offer an optional online questionnaire in about a dozen languages. We also, all of our reports, of course, as you know, are available in about a dozen languages. So we can report in all of the most important languages of the world, essentially, with the exception of some of the Asian languages, which we haven't done yet. And the questionnaire allows people to anonymously, and only link to their sample code, and only the physician knows who the patient is, to tell us about lifestyle factors and other things in their lives. If they exercise, if they have hepatitis, if they have high glucose, if they suffer from insomnia, if they have high blood pressure. If they don't complete the questionnaire, this page doesn't have any highlighting. You don't see those blue boxes, but you just have the information there. And then in the PDF of the questionnaire, each of those bullet point articles are links to clinically published peer-reviewed articles, the most important ones, because there are thousands, that show why this factor is influencing in the aging process. So you can click on that and go and read the abstract and see that there is strong science behind these factors, and this is why it's important to address these factors in your life if you can. If you have filled out the questionnaire, those factors that you've responded to are going to be highlighted in blue. And so this is a clinical interpretive guide, if you will, for the physician and for the patient to see what factors, family history, genetic, lifestyle, are influencing that individual's rate of aging, and look to address those factors if you can. And I would say that over 95% of all of our clients do the questionnaire, because we work obviously with people who are really keen to make a difference in their health and to adopt healthy lifestyles, and they want to understand what are the factors that are driving their rate of aging and make changes if they can. And finally, just to conclude here, this is a really easy test for you to use. First of all, we provide everybody kits, depending on where you are. If you're in Europe, we provide kits directly to you. We work with overnight courier company, TNT, to get those kits back to our lab. All of this is included in the price, the shipping, the kits, everything. We have a great customer service and logistics team that supports our physicians throughout Europe. In the United States, we work with a very strong lab partner. We also provide kits there. If you're in other countries around the world, we have lab partners in many, many countries. We work now in approximately 40 countries around the world. Patients don't have to fast. This is also a question, often a question we get asked. Do you have to fast? The answer is no. This is not a glucose test. You don't have to fast. Diet makes a big difference in your telomere length, but not what you had for breakfast. It's more of a long-term thing. So fasting is not an issue. We only need 1 tube of blood, real easy. No spinning, nothing to be done by the physician or the nurse team. You just draw 1 tube of blood, which comes in the kit, and return it to us. You have this anonymous questionnaire that we talked about. This is the website where you can see it. If you haven't worked with us, if you haven't done your test, if you want to see the questionnaire, let us know. We can give you access to it with a temporary code. But it's really very intuitive, and I think people find it very easy to do. And then our turnaround times are typically, we're conservative here. We say 2 to 4 weeks. Often we turn around reports faster than that. Depends a little bit on where the sample is coming from and how often we ship to our laboratories here in Madrid. But we've really gotten very good at turning around reports quickly. And all of our reports come on a secure online platform. We don't email reports. We are a US HIPAA compliant laboratory. We have the equivalent compliance and data protection in every country in which we work around the world. And so you'll get an automatic email from us telling you when a patient report is available, and you can go online and download that report in PDF. And similarly, if the patient has done the questionnaire, you can download his or her questionnaire. And really, doctors, I like to emphasize that doctors like this because sometimes they find out information that the patients have not themselves told them when they go through the questionnaire. So that's really the presentation that I wanted to do today. I hope it's not been too long, and I hope you found it interesting, and I'm happy to take any questions that people may have.
Thank you very much, Steve. Just as a reminder, please use the chat box on the top of the screen to write your questions so that Steve can proceed and answer them for you. We will give you 2 minutes so that everybody can write in their questions. Thank you. Thank you very much for your questions. We're going to do 5 to 10 minutes to answer some questions, so feel free to keep writing your questions as Steve goes ahead and answers them. Thank you.
Okay, so we've got quite a few questions here. I won't be able to address all of them. If I don't address your question, do note on the screen our email, info@lifelink.com. Feel free to write us with the question if you don't mind, follow up with us, and we'll get back to you in a personal format. We'll either write back to you or we can have a phone call to address your specific question. The questions that I want to answer, let me start here. First up is about pricing. People would like to know a little bit about what do we charge for the test. We have made a big effort in the last few years to bring the cost of the test down very significantly. The cost of the test depends a little bit on where you are geographically, because the shipping and the sample handling is a significant part of the cost of doing our service. In Europe, we provide, in the EU, our test to physicians normally for 249 euros, with a recommended price to be sold to the patient for 349, plus the consultancy fees, the interpretation fees that the physicians wish to charge. In the UK, I believe it's £199, which with the exchange rate is roughly similar. Maybe it's now changed slightly, but it's a very affordable test in Europe. In North America, the test price depends on the volume of testing that physicians do with us. We also offer some prepaid options, and the price range in the United States and North America is between $299 and $395. And so those are the main prices. If you work in other parts of the world, if you're in the GCC or you're in Asia or other markets, contact us and we can give you the specifics. Second question I hear is, how do I address, bad results? What do I specifically tell patients to do when they don't get satisfactory results? First thing I want to say is, if you don't know this, we have a very extensive library of CME level materials to educate you on telomere biology, on the clinical use of the test, and what to do in terms of therapeutic interventions. So I highly encourage you to go to those resources, which we make available free to you. You can get them through our website, lifelink.com. We have a Dropbox where you can download presentations. We have 3 videos available for people. 1 to explain the uses of the test to the patients. 2 for physicians. 1 is, what is the clinical utility of telomere testing? And the other is, how do I interpret the test results? These are videos developed with Dr. Mark Tagger, who's a very well-known physician in the United States in the area of preventive medicine. And you can watch these 3 videos. Literally, the total running time of the 3 videos is 15 minutes. And we have these fantastic presentations that you can download and read. And they're very pragmatic. And they go through specific recommendations around exercise, diet, supplements that have clinical evidence, that have got underpinning of real science behind them, that you can use to guide your patients about things that they can do. The important thing that I need to highlight to people is, this isn't a test to determine if you're going to give somebody a statin. If we think about lab tests, a lot of lab tests today, the response is, oh, you have high cholesterol? I'm going to put you on Lipitor. I'm going to put you on Crestor. The physicians that we work with and the patients that we work with are people who don't want to take Crestor. They don't want to get on Lipitor. They don't want to be on lifestyle drugs that try to mitigate the symptoms but don't address the underlying causes, that they have a terrible diet, that they're obese, and that's why they have high cholesterol. Our goal as a laboratory is to support our physicians and our patients to avoid the necessity to use prescription drugs to mitigate the symptoms without addressing the underlying causes. There is no pharmaceutical drug today that you can write a prescription for that's gone through clinical trials that will rejuvenate telomeres. I've discussed what exists today. Now, there are many companies that have supplements. And in one of our presentations, we go through all of the companies that exist that have supplements and provide you all the information, a wealth of information about who's out there. And then you can use these presentations to go and look at the different options and determine, I think it's pretty easy to see, who are the companies that are more credible and more scientific and that you might want to consider using. But in these types of wellness programs where we're focusing on optimizing people's health, I always want to emphasize that if you don't do the basic stuff, if you don't take care of the diet, nutrition, if you don't do some kind of exercise, if you don't manage stress, if you don't manage your alcohol consumption, if you don't sleep correctly, the supplements aren't going to correct those core issues. For me, it's like concentric circles. The first circle is all the things that we all know we should do. And I sometimes hear doctors cynically say, well, why do I need to do your test to tell my patient to work out more or to lose weight, because I know that already? And I answer is what you don't monitor, what you don't measure, you don't do. At the end of the day, people respond to objective measurements that give them insight to how their health is evolving. And that's the point of using screening tests. Because if we didn't do that, then people wouldn't know where they stand. So, and very few people, by the way, as we all know, are able to do these kinds of things that, yes, we know conceptually we should do, but we don't do every day. So you do that core set of things, then you move on to the supplements like omega-3s and vitamin Ds and other things that most of us need. And then you move on to telomerase activation supplements and more, let's say, expensive and perhaps less widely accepted solutions that you need to evaluate on a case-by-case basis. Let me see some of their questions here. So there's a question about one of the companies that manufactures supplements, which is called TA Sciences, and their product TA65. So TA Sciences is among the very best companies in the field of telomerase upregulation, if not the best. They are the company that's done more scientific research around their product. They've done a major double-blind, placebo-controlled study, which they can facilitate to you, which they published in a peer-reviewed article that looked at the efficacy of TA65 in 100 individuals over a year. And the outcome of that study is pretty positive. And I would encourage people who are interested in looking at this to contact TA65, contact TA Sciences, excuse me, request the publication, which they make available, of course, freely to anyone who wants it, and evaluate the appropriateness of this product for a particular patient. We are a clinical laboratory. We don't have patient histories, and we obviously aren't in a position to make specific recommendations around any supplement, whether it's even a vitamin, because we don't have access to that information. We support our physicians, and then it's up to you to evaluate the requirements for each individual. Another question about testosterone. Does testosterone influence length? And does hormone replacement therapies work? And of course the answer is yes. Hormone levels are very important in telomere maintenance. And low levels of testosterone or estrogens or progesterone do affect our aging process. And so what we see is that in many of our physicians, the program of BHRT, bioidentical hormone replacement therapies, is becoming more and more a standard tool in their arsenal, together with the supplements, to really put people into their optimal levels of hormones and optimize their health. Of course, this needs to be done, needless to say, under careful physician care and with frequent and appropriate testing to ensure that whatever hormones you take are the appropriate levels. Let me see one more question. Supplements or foods that have been shown to slow down the aging process. In our module 3 of the 4 modules that we have for training for you, we go through the specific foods, the superfoods, the green leafy foods, all of the specific foods where there is a good scientific literature available for you to be able to recommend the real clinical basis what foods people should focus on eating and not eating. So yes, there's a lot of evidence on that, and I would encourage you to go to our Dropbox and you can access all of this information on the specific foods that are out there. There's another question about the pharmacological drug and whether it would affect healthy cells. And the answer is it wouldn't affect healthy cells because normal healthy cells don't express telomerase. So in a normal person, this drug is only going to impact the cancer cells because those are the cells, these are the cells that are expressing abnormal levels of telomerase. So the drug, and I think the drug, I don't know all of the molecular pathways by which the drug works, but I think the drug is designed to have a binding to specific proteins found on the particular types of cancer that they're seeking to address. So the interaction of the drug is tailored to go to the cancer cells themselves. Okay, well, I think we're coming up or a little past the time that we had allotted. I'd like to thank everyone very much for their interest, and I hope this has been helpful. And we'll be very happy to follow up with you individually. I believe we've also recorded this session. If you have colleagues or people who weren't able to attend, we'll be repeating this today, later in the day. In the emails that we sent out, we gave you the times depending on where you are. And I remind you that we have our website at lifelink.com, and our general email address is info@lifelink.com. And I'll also mention that it would be great if you're interested in telomere biology, follow us on social media, on Twitter, on Facebook, these things, because we're constantly updating things. We've got an active blog, and you can even go and visit our Oncocheck website for our cancer study. That's on www.oncocheck.eu, and that really tells you a lot about the studies we're doing in cancer. And you can also look us up on YouTube. So lots of things that we're doing to keep people informed who are interested about telomere biology and its advances. Thank you so much.